webinar. And today, it's pretty exciting to have some of the pioneers of computational chemistry and prediction on the webinar today to let us see the storied history of these kinds of processes. Target 2035 is an intent to identify a pharmacological or small molecule modulator for as many proteins in the human genome as we can in 15 years. And currently, it's way too expensive to do that, and it's almost sort of fantasy to think about doing it. It must transform from an experimental science to a computational science. And part of the strategy to get there is to take the progress that's being made in the field and compare it so that as a community, we can identify best practices and get us to that end game faster. I mean, it will happen that we'll be able to predict small molecule binders, maybe even leads or even clinical candidates for every protein. Whether that's 50 years, 30 years, 10 years, or 5 years from now is the question. And we need to get there as fast as we can. So the purpose of the three talks today is to say what's happened in the past by the folks who are instrumental in developing the methods and in putting together competitions, maybe not competition is the wrong word, but sort of benchmarking exercises to compare and contrast methods, and then to look to the future as to how we can do that and couple it more tightly with experimental methods. And that's, so there'll be a question and answer box at the bottom, because I'm sure folks will have lots of questions as this goes on. So you can see the Q&A box down at the bottom. Please feel free to type in your questions, and maybe some of the presenters can answer them online. And if not, I'll bring them up. And so for the talks will be organized in a set of three, and the first will be from Ruben and Max from San Diego MallSoft, where they'll present the perspective from some participants and some experts, and they're really good at what they do, and they'll be able to say their experience in these kinds of competitions. David will speak about how these things are organized and what are their challenges in putting them together and measuring their output. And then finally, Mathieu and Alex will speak about the next generation of these kinds of competitions that is tightly coupled with experimental science, so that we can actually proactively test predictions. And so with no further ado, I'll pass the torch on to, I think, Max, you're first, or Ruben, well, you guys can arm wrestle and see Ruben who goes first. Okay, and then we're really excited to hear, and thanks so much for taking the time early on this morning to give you your seminar. Thanks so much. Okay, over to you, Ruben.
All right, thank you, Al. So do you see me? All right, so it's shared, right? All right, so thank you, Al, for the introduction, and thank you for and you and Mathieu and Cheryl for organizing all this. So I was a bit confused about what the topic is and what the perspective, but so it is. I understand that it's just the impression from the first competitions or assessments. I call it evaluating 3D prediction tools, dreams, and reality, and put a random picture with some ligand. I can, yeah, as a picture, just to as a good morning. Okay, now let's. So they basically, so I will mostly talk about the first few CASPs and then Capri competitions, which are protein docking competitions. And we started doing that because 94 was a very exciting year for us. And Max and I will published a few key papers on the global optimization of structures, which allow to basically take a system with any number of molecules, not just with one molecules, and globally optimize it, basically sample the whole space. And you see the recording. So this is the video from that time. This is from what is that, almost 30 years ago, 25 years ago. So you can see that you start an arbitrary point, and then the sampling and optimizing the energy of a system which is described in internal coordinates. That was the key. It is searching for different states, and then at the end we find the global minimum, and then we look at all the good confirmations collected, and then the system had these features of you describe the system as a full atom system with the continuous movements. It's realistic. It's not a 9 points in a cube and you shuffle the points in a cube. So this is the global optimization internal coordinates. There is other local minimization steps there, and then there is it uses the information from available structures to make the moves, and these are large moves. They're not small moves, they're large moves. And there is a kind of quite elaborate global optimization logic, which includes the reactive history mechanism. We basically learn from what you've done before, and depending on that, either search certain parts of space or not. And so we published some papers, just two of them, but it was a large number. It was all in '94. So then at the same time, what happened is that we moved, Max and I, we moved from NYU, from Heidelberg, where like this version, which we and we still use until now, the of ICM was written. We moved to New York, to Manhattan, and then started working at NYU, and then at the same time I started MALSOFT in '94. So basically, at that time, this competition started. I actually don't remember I don't even remember if I participated in CASP 1 or not, but definitely CASP 2, and you can see this quotes, this 96 paper, and it's a good example of how CASP 2, which was an Asilomar, very nice, meaning how it went. So here my comments are the confusion, because it looked like a feeding frenzy. So this is the describing how the participants, at least some of them, felt. It was, from my humble point of view, it was extremely poorly designed because of this competing interests and God knows what. And everything was obscure. If you actually read the paper and try to understand who did what and why and what are the results, you'll be confused, because it's many different sections. It's not clear how things are evaluated. This is, at that time already, there were different categories for not only, well, there was first introduced Dawkins category, but it was also homology modeling or comparative modeling, fold recognition, ab initio, and some other, depending on the situation with the previous knowledge. But in the ab initio category, given a sequence, predict the structure. Now, with much larger number of structures, it's less interesting, because you can just copy things from the existing structures in most cases, or at least you can copy some information. All right, so then basically, I don't have the, well, we did well in those competitions. In each category, the paper, which was published by the organizers, and it was largely a political document. It's hard to figure out in terms of methods what was there. Mentions us in each category, and the results were pretty good. And then, but we were quite disappointed. I will explain later why. So then the competition on protein Dawkins became very important for us, because in '94 we also published this paper in '94, where we, for the first time, show that we can take it two proteins and at least one of them in a non-bound conformation, in a different conformation with side chains all being different, and we can sample the whole space to predict how the complex is formed. And this paper also showed that after we generate this set of solutions, then we can refine them with the full flexibility of the interface, and the correct one splits out. It's separated by considerable energy gaps. So it basically allows us to recognize the correct one, and it is indeed a correct one as well. So that gave us a good ammunition for what was the early. It was not at that time. It was not Capri, but that was the first competition from Natalie Strinatka and many other good people. So that's our recording that made at that time for protein docking. This is a refinement step, which you see now, where the interface is being refined, and then the good solution shows itself more prominently compared to the incorrect ones, which cannot improve the calculation as much. So with that, we participated in that competition. That competition was easy, so everyone got approximately the right structure. The numbers were comparable. But then there were difficult competitions, and then Capri 2002, we improved the protein docking. We introduced the grid energies at that time, so here the procedure became more sophisticated, and also the objective was sophisticated. We also introduced the benchmarks with uncomplex structures, as well as structure of the complex where there's induced fit, and refined before the competition. So when the competition came, we were well prepared. And then this is one of the targets there. And then we participated in, I think, 5 rounds of Capri, and Max will correct me if I'm wrong, because I couldn't find a single presentation from that time. So you can see, so now here we need to sample a huge surface of hemagglutinin, and you can see that the sampling is quite comprehensive. It doesn't know where the answer is, and it tries everything. And so the results were indeed good. So we were the best rank in each of those competitions we participated in. So that's the overall sampling everywhere, and then it basically finds the good one. And then after finding the good one, so this is the paper where the method and some of the results are described. And what you can see here is that the refinement not only places it correctly, but also it, let's see, the green side chain moves into the correct one. And so the side chains after optimization move it in the correct one. And for this complex, you see also the correct solution is the lowest energy. That's another important thing, is that how you define that competition. Like, should it be the lowest energy, or should it be just, anything within the top 20,000 solutions? And at that time in these competitions, only in Capri, partially under our pressure, the rules were better defined. They were much better defined in Capri than in CASP. In CASP, it was just a total mess. So these are the results for Capri. To some degree, you can see the red and green. This is correct and predicted. And of course, the answer is unknown, but then the prediction matches it pretty well. You see that by our summary, we're well scored. We're on top of this large number of teams, and this is some other rounds of Capri. You can see also how the prediction goes compared to the experimental structure. Another good thing in Capri is that the evaluation was much more reasonable, because the comparison measure was well defined, and the number of submitted solutions was well defined. So this is our 3 to 5 rounds evaluation. We're on top. This is not alphabetically sorted. This is just we got the better overall summary. So the CASP. So now let's go to the. I think it's time for me to finish, right? So the what are the problems with the early CASP? Well, the rules not defined before submission, right? You submit and you have no idea if you submit one structure and somebody else submits 10,000 structures. They don't tell you if they're going to look at the top 5, top 1. So the it's not clear who and how will evaluate and declare the ranking. The evaluation measure, and that's the most important thing, well, sorry for repeating it 2 times, it's just how important that is, not defined before submission. And the judges who declare the ranking and present comparison, they also participate in the competition themselves, which is also weird. The judges, so how they appoint, like the order, and then who is given the honor of publishing a paper in Proteins next to their assessment is not clear. So obscurity and bias in evaluation and the publication of results and lack of automation and transparency were striking. So, and I came to John Molt, I said, John, it doesn't, like, it's just wrong. You know, you need to be transparent, you need to declare, have more tight rules. And he said, well, sorry, it's very political, And I said, okay, goodbye, never to return. So
So Ruben, you're at 12 minutes. I don't know how long Max is supposed to speak,
right,
but you have
he's
20
also
between
10
you.
minutes. Yeah, we started a bit late. Yeah,
You're at
right.
12.
So this is a fascinating story for after that, but that's how cheating was going on in CASP. But now you will not learn about that. But it's a very interesting story. And then with that, this is my dream slide, last slide, for your competition and utopian project, how it should be, right? Well, it should be universal set of rules and measures. And then each new soul structure, SGC or PDB, is a new task. And then the evaluation should be done by automated procedure, and the submissions for each task should be defined. Like for ligand prediction, for example, it could be 1,000 or 100, but then for some problems it should be just one. Give us your top solution, and the transparency. These are the rules which should exist. With that, I think I just want to thank you. And this are the members of the team, of different teams of CASP and Capri at that time, which was 20 years ago. And thank you for organizing and running that. Okay, so I pass that. I stop share.
So transparency, I think, was the main message.
Right, and well-defined rules, right? So
exactly.
you need to, like,
clarity.
if it's a competition, just tell us. If you measure the time, or you measure how you jump, but you start running, turns out that you're supposed to jump up. And
fair enough. Okay, all right, Max, over to you. It's not up yet.
Anyway, I'll just be quick, go through the our participation in D3R organized grant challenges. Well, first of all, what is D3R? That was a drug design data resource created by Romy Amar and Mike Gilson at UCSD. The idea of this D3R was to facilitate release of proprietary data from mostly pharmas to the broader community, and through that actually help along multiple lines. But one of the activities that they envisioned and actually accomplished was the so-called grant challenges, the blind challenges, where the participants would be invited to predict structures and activities of compounds. And there were 5 challenges total. We participated in the last 3. Typically, a challenge would include 1 or 2 targets, usually of high importance for drug development, a few dozen X-ray structures, maybe 100 of activity data points. In this last 3, there was FXR, you see some kinases, cadepsin, so representatives of multiple classes of targets. The range of challenges was to predict poses, primarily in cross-docking, so the more realistic mode, but also there was a sub-challenge for self-docking, which is useful for benchmarking of methods. There was activity prediction, primarily with ranking of multiple ligands on the order of 100, but we also had a sub-challenge for smaller sets where quantitative predictions were made, ideally absolute values of binding energy. There were representatives of targets that had sort of common difficulties that we often see with modeling. Let's say there was a highly flexible target of XR, which has all this flexibility around the pocket. I try to show here how if you superimpose all the PDV structures, you get this kind of spaghetti because there's so much movement around the pocket. And there were open pockets, which is another common class of difficult targets, proteases in particular. In this case, it was catepsin, had this kind of wide open pocket with shallow structure. I have to say that all pretty much targets were pretty well studied. So that is sort of maybe a limitation of what we got from these challenges, was that we were always working with something that already had dozens of structures and lots of activity data points. Now, what do I perceive as benefits, at least on our side, from participating in this? One thing was that it was really stimulating. We had a number of, sort of techniques that were sort of in an embryonic state when we got to one or another challenge, and we felt that it was useful for a particular target. And we actually, during those 1 or 2 months that we had to submit the predictions, we could sort of improve on it and make it usable for a particular challenge. There's at least a few things here that we developed, or well, we developed before, but really made it to work during the grand challenges. One of them was this hybrid QSAR predictions, which combines the more knowledge-based and chemical phenomenological potentials with physical interactions together. And it helped us to get better predictions. In multiple receptor conformation dockings, we added this offsets that helped us make the procedure recognize the correct pocket conformations for a particular ligand. And again, we did have already the procedure that handled microcycles, for example, but during the challenge, we made an effort to actually benchmark it more extensively so that we know what particular parameters need to be used for this kind of systems. Again, it helped us in getting good results. Now, next thing I wanted to mention was that well, every competition of this type of course will run into metrics issues. Ruben already mentioned some of that in his talk, and I want to show you an example what happened in some of the grand challenges. We had this post predictions, right? And a sort of natural measure is RMSD, the commonly used across the entire field, right? And well, the mean is one way of combining RMSDs across the set of target structures, right? And you can see, for instance, on the top row of this plots that, well, the red arrow is our group's results. We did pretty well with those means. Now, well, some people felt that, and it's not wrong, that mean RMSD includes RMSDs for ligands that had 6 or 9 angstrom, let's say, deviations, which are essentially all wrong. But if you have a 6 versus 9, you might have significantly lower overall average, which may be kind of a noise rather than meaningful difference. So at some point there was a switch to well, medians, right? Median is sort of restricting the effect of this kind of issues with high values. But on the other hand, it just throws away basically half of the targets, target set, because if you just predict, 51% of easier structures, well, then you can get a very good number, even if the 49% were totally mispredicted. So, there is no perfect metric there, but you can see that, for instance, for us, with one metric we did very well, and with the other, well, we were somewhere in front of the line, but significantly less impressive looking sort of positioning, at least in the list. Briefly mention another issue was that multiple Submissions were allowed, so I think, okay, 1, 2, or slightly varied parameters are okay, but some groups submitted, like I'm showing here, 7 different submissions, at which point it's not really clear. You could just can you actually tell which of your methods work and which don't, or are you just throwing in stuff there and hoping that one of them will come out good? I think this has to be, thought through when you design this kind of competitions. And of course, there are some issues just related to the experimental data. For example, when organizers evaluated the activity data, at some point there was a switch from including inactive compounds data to splitting them into another sort of sub-competition where just active versus inactive was predicted. And again, as it happens, if we include, the inactives, then, for instance, our results were, say at the top, and with the switch we suddenly moved somewhere. So these sort of things need to be thought through. Also, how do you deal with certain things? Maybe in some cases just multiple metrics is the only way, but there should be some agreement on what is the metric before the competition is run. As a sort of curiosity, there are also things with post predictions where just limitation of crystallography, let's say, is that sometimes pose is defined not just by interactions with the primary target protein, but the neighbors in the crystal. And we felt that some of the deviations from experimental structure were not really reflecting anything. So we really have a solution, but rather just yes, sorry.
You guys are at 25 minutes, yeah. So
okay. Just 2 slides, I think, and then
okay.
So back to the, with all the issues, the benefits, I think, far outweigh the problems, and the competitions, I think, need to keep going. I feel that the stimulation of development is great, and also, as participants, we got to publish things in the special issues that were followed. Each competition was followed by a special issue of Journal of Computer Molecular Design. There were evaluation meetings, also excellent meetings. I think was great to present and meet other people and discuss all sort of interesting things. And now, just what we learned from this, maybe one thing is that, again, I'm showing our results and they may look sort of good as far as ranking, but think about it, the taus, Kendall tau, are at the level of below 0.5. So, it's great to show that we were up in front of maybe other groups, but I couldn't really think of showing this to a chemist or a biologist and with a straight face saying that less than 0.5 tau is a great prediction, especially if you consider that, again, I mentioned this already, but these targets have like thousands of data points available for them. So we have a long way to go with affinity prediction, I think. But on the other hand, docking, actually, many groups did well, we did well, and that part of the enterprise is really maturing now. The affinity prediction, a lot to be done there. And I think one of the problems why we see this huge drop between what you get in blind competition versus sort of things that people typically report in papers when they benchmark their methods is that very kind of lenient measures are used, like leave one out consolidation or just random subsets. We found that more strict, stringent cross-validation, like taking clusters and then separating, let's say, a third of clusters out, that's when you get something comparable to blind prediction. Other sort of low kind of things that they really don't show what happens in real life, and it was useful to see in the grand challenge what the test set should look like. And well, I just want to acknowledge Paul Lamb, which was the person at Molsov that was actually running most of the predictions for grand challenges. Yeah, thank you.
Thanks so much. That was so interesting to learn about I think the clarity, again, the same message, right? Clarity of what we're asking for and a transparency in how it's assessed is a really top thing. And then it can be useful for the community. I love to see how all the science that you learn from participating. So thanks so much. And we're going to go over to David now to talk about how one of the challenges was run, both, and we're going to learn about the science and potentially also the organization.
Yeah.
Okay, over to you.
Can you see my slides okay?
Yep, perfect.
Okay, great, thanks. Yeah, I'll try to go, I agree with a lot of what's been said already. The sample challenges have been partly include a physical property prediction component and sometimes include binding. So for example, when D3R hasn't been running, we have worked on a binding component occasionally, and we're getting ready to run a couple more challenges in that area. I want to really focus on lessons we've learned, and some of those that we've already heard, I agree with a lot, like clear metrics. These challenges, I don't want to give too much background, but these are, crowdsourcing challenges focused on driving progress in the area, and they include physical properties, typically host-guest binding, and sometimes a protein ligand component. One of the things that we've observed with protein ligand binding is that sometimes people just do badly, and the main lesson is something like, this was a really hard target. And you'd like to be able to learn a little more than that all the time. So sometimes you can benefit from, or an ideal challenge might not just have a protein ligand component, but might have related other challenges. For example, for the types of compounds you're predicting binding for, how hard is it to predict the pKa? Maybe you're doing badly on predicting binding because you're getting the pKa's wrong, or you're not able to even predict simple things like how well your molecule partitions between polar and apolar environments. So sometimes simpler challenges can provide important insight into why hard challenges like binding can fail. But today I'm going to focus mostly on things we've learned that can help challenges like what you're envisioning. One of them is that protonation state prediction is hard. This graph in the middle here is calculated log D versus experiments, and the X's are the original predictions somebody made. I'm sorry, the red are the original predictions, and the X's are what happened after they did a better job of predicting pKa's for the compounds retrospectively. So important lessons can come out of this. Also, though, over time, we've been able to see a progress in how well people can predict certain properties, for example, on solvation free energies, till they get to the point where they can sometimes predict when the experiments are wrong. We can sometimes now detect when an experiment needs to be redone because many of the participants will agree on a value that's very different from the experimental one. And so we like seeing that kind of progress over time. We want to see that same sort of thing happen with protein ligand binding. So just to highlight a couple of lessons there have been in that area. We ran years ago a virtual screening competition on HIV integrase fragment binding. And one part of that challenge was to predict actives versus inactives from a set of compounds. And this is pretty challenging. This is binding to a symmetric dimer, and there's actually 3 different binding sites that these fragments could hit. And it turned out that the best set of the group or method or person who did the best on predicting which compounds bind here was a human expert with 10 years of experience working on HIV integrase binding. So they more or less, they used some computational methods, but then the final step in the process was to look at all of the compounds and decide what they thought should bind and submit those predictions. So that's a method, and that was the best method here, but it's not a method that most of us have access to. So that's an important consideration when staging these challenges. You have sort of an opportunity to decide, what do you want to learn? And do you want to allow like human experts to participate or not? Most methods in this challenge did no better than random. So this is sort of a more typical enrichment plot. And the best automated method was a binding free energy calculation method that used millions of hours of GPU time. And the largest reason they failed, aside from running out of GPU time because they couldn't finish the calculations, was when they had incorrect initial binding modes. There's also a posed prediction challenge as part of this. And we gave a hint that most of the compounds bound in the ledge F site. It ended up that 50 out of 55 compounds bound in the ledge F site. And most of the participants didn't use the hint. So this is a graph of looking at how many methods place the compounds into the correct binding site. And of those who didn't use the hint, only about 2 got more than half of them into the correct binding site. So average RMSD was very bad. This is mainly just to say these are still hard problems. So design challenges to focus on what you want to learn. Many different factors go into overall performance aside from how many actives you find in the end. So for example, database curation or pre-filtering will affect success rates. Like if you just tell somebody, here, screen all of enamine and we'll take the most promising compounds, they might give you predictions of a whole bunch of compounds that are clearly very unexciting as drugs because they're too hydrophobic and gonna have solubility problems or something. And so you decide, oh, okay, well, we're not gonna test those because they're obviously bad. Well, you could have made that decision to begin with. So you're just gonna learn that people are bad at filtering databases to throw out hydrophobic stuff. Protein structure selection, prep, potentiation state selection, that's hard. Ligand preparation and curation can be hard. Knowledge of what experiments you're going to do, not everybody's going to know that or understand what you're going to be able to measure. Not everyone's going to have expertise on the target or the literature relevant to that target. We just heard about how there's a whole bunch of literature compounds available for cathepsin. Most participants probably didn't have that and weren't looking at it. Interpretation and use of prior experiments. So usually no single group has experience in all of these areas. So success is going to require knowing and doing well on all of these. Most people are going to fail. So in a recent FIP2 challenge in SAMPL, most participants picked compounds that were too insoluble and the experimentalists decided they weren't going to assay them because they clearly were bad. So the end lesson was computational people are bad at picking compounds we want to assay, which is not the lesson they intended to learn. So you probably Case probably wants to pre-filter databases to make sure that the compounds people are predicting are compounds they would want to assay, or make assay selection criteria known. Ensure that participants know what they're being tested on. Are you being tested on library screening or also their ability to filter? If you're judging hit rates, some participants are going to increase their hit rates probably by using target knowledge like pharmacophore filtering prior to using other methods. Is that what you want? Is the highest hit rate really the best method, or does it mean somebody did the best job filtering their database? So there's a lot of things that Case will need to assess carefully there, I think, to decide what people are really being judged on. One of the lessons from one of the grand challenges was essentially which protein structure you use is important, which is something we know. Groups who invest more effort in picking good structures for computational methods are going to perform better. Is that what you want? Or should you maybe provide recommended structures for people to use, or a range of structures? Some people aren't going to carefully check the literature. They may just pull the first structure of that protein from the PDB and use it without much thought. If you wanted to try to isolate more just performance of the method, you could involve target experts in protein prep or provide pre-prepared structures from high-quality methods, or give a diverse range of prep structures to provide a range of possibilities so that participants can try their methods using multiple structures and then make more educated decisions. And then also you're going to want to tell people, probably want to tell people who are participating, here are some things we know that are going to be key important decisions you have to make about this. Otherwise, modeler expertise and amount of prep effort is going to end up playing a huge role in success. So these kinds of things, how much you want to test human expertise versus inform people so they have a more level playing field in terms of expertise, will play a big role in the outcomes you see. I know we don't have much time, but I just want to say pKa prediction is still really hard. And at least with some methods, it's going to end up impacting performance a whole lot. I won't, in the interest of time, I won't go through the graph, but maybe you want to think about pre-preparing some libraries which enumerate likely states, for example. And my slide is not advancing very quickly here. Okay. Also, submissions. People are really bad at following instructions on submission formats. So the required format for this challenge is on the left. What they actually submitted was on the right. This is an example. So normally we run some automated validation scripts to make sure that we'll be able to parse the submissions, because if we don't, when we go to analyze, we find we can't analyze most of the results because people violated the formats. But this is an example that actually passed our automated validation. They just included an extra column in a CSV, but then a human working on it had to change their logic in order to correctly handle the format. Usually our automated submission validator is kicking back about 50% of submissions because people aren't following instructions. And so that's 50% of submissions we don't have to fix, we're making the users fix. So highly recommend automatically validating things, otherwise you have all kinds of problems. Then metrics. People tend to obsess over who beat who by 0.01 angstroms in mean RMSD or something. So in SAMPL, we usually deliberately use multiple performance metrics. And that tends to mean there's not a single winner unless one method is better in every way or in many ways. And then also do proper error analysis. You need error bars on your metrics so that there's no clear winner unless there's really a clear winner. And then I would say focus more on rewarding people who learned a lot and improve than on rewarding the people who win. So maybe when you're thinking about picking talks for meetings or something, pick abstracts not based on like best performance, but biggest lessons learned, because you're after driving progress, not just having people race. And people are going to start tuning methods for a particular target. We routinely see empirical corrections. This is a method on host guest binding where they had adjusted based on literature data. So you want to think about whether these types of methods or machine learning methods are in a separate category or how these should be handled. So I'll stop there because I know we don't have much time, but I hope some of these lessons can translate to your challenge.
I found that incredibly useful. And I know that you've been helping us, and Alex and Mathieu have been thinking about all the things that both you, the three of you have said. So this is fantastic. So why don't we go to Alexander and Mathieu, and they can describe how they've been taking all your learnings and hopefully coming up with something that at least gets close to what you guys would think would be a proper way to run one of these sample or challenges. And I think, so Alexander, I see both have come on, and I'll let you guys take it from here. And there's a question for you, David, in the Q&A. Okay, over to you guys.
Yeah, thank you very much, Al, for inviting me, and you already set the bar very high. Thanks for the many good tips here. I'll try to share my screen. That's visible?
Yes.
Oh yeah. Okay. Good. Great. So I'll talk about cash from a pharmaceutical company standpoint and how we initiated the whole thing, and then I would like to turn over to Matt to present his further insights. I'll skip over that. This is just introduction. Next. We saved some time, so sorry for that. Next, let's go over that, and over that. So I mean, cache is really designed based on what you have learned and what CASP has done in the past 27 years, and that was really cool to see Ruben's experience with that, and we really took that and tried to do something similar with respect to hit finding. Next slide, please. So in 2020 in November, Al and Christina, and I was also involved in that with a number of colleagues. We had a larger meeting with 80 scientists and funders, and the goal was to prioritize different area of computational chemistry that might advance more rapidly if organized as a community-wide effort. And we pitched 3 topics. The one of was quality of hit probe identification. The second, improved ADME talks and developability models. And the third one, quality of hit to drug. So the optimization part of drug discovery. And there was a lot of input, dozens of pages of chat protocols that were analyzed, especially by Christina. And from that, we got hints on the prioritization on the one hand, and also the cache concept evolved. And we said we would like to focus on the quality of hit to probe identification. And next slide, please. So from that, as said, the critical assessment of computational hit finding experiments was coined, and so CASH is a benchmarking initiative to reveal the most efficient computational methods. So it's primarily on methods, a member-driven public-private partnership, and we strive for unbiased, high-quality experimental feedback. So we really want to synthesize and test the compounds, as you will see later on in the slides. The benchmarking effort is to identify small molecule leads and the validation of hit finding algorithms. And all the data will be generated in one central hub with the SGC. And of course, all data will be made publicly available without restrictions. And computational algorithms can remain proprietary, but of course, it's rewarded if people really describe and publish the detailed methods. And publication and participants who had elected to be anonymous will have the choice to disclose and identify their identity at any time. So the next slide, please. So this is on the target selection. So we aim to provide 3 challenges per year with 3 different targets, and targets will be chosen on the one hand of biological insights. We really want to do this with very interesting, currently interesting targets, and don't want to go back to the old stuff. Of course, a selection of the targets is influenced by meeting technical requirements. And of course, we would need to have a availability of cost-effective assays for those targets. And with respect to the challenges, we see 5 different types of data situations. 3 of them where we have a protein structure, crystal structure, or whatever available. And no structure available, where we only have SAR, and here are the details to that, but I don't want to go into detail here as we are short of time. Next slide, please. And this is a workflow, how the CASH challenge will look like. First is, of course, the hit finding challenges, as with different types of SAR or protein structure available. A second, of course, important, the libraries, which libraries we want to use for virtual screening, and on the other hand, of course, also synthetic realization. And our most important library here, of course, is the enamine library, real space, and the sync library. And just as a remark here, I mean, today is really a black day. The enamine library is made in Ukraine, in Kiev, yeah, and I just can't not make this comment that the problems we have at the moment are not so far away. Yeah, this will of course impact what we're doing here. Third point, participants will predict, on the one hand, derive predictions for small molecules to be synthesized. The compounds will be sourced and synthesized and then tested. and funneled and prioritized. There will be a first round of analysis, and we'll go into a second type of optimization here. And the plan is that the participants can submit 100 compounds with the first round of proposals, and then another 100 with the second round. And at the end, all the compounds and assay data will be made available publicly, and all screening data and also the virtual compounds are then in the open. Next slide, please. So these are for example, the 3 challenges: protein target 1, 2, and 3. And here you see how long the different steps take for these 3 targets. The overall duration would be 24 months. And for the first protein target, you see there is a time of 2 to 3 months for predictions, then 4 to 6 months for compound logistics screening, 5 to 7 months, and the second round of predictions and compound logistics, and final 3 months for the assessment of and data release. So overall, the process of one protein target would take 18 months. And that is planned. Next slide, please. Here, these are the proposed assessment criteria for an arbitrary challenge. We are working on the detailed challenge criteria for each and every target. These might be not identical, so we'll customize these. And this is one proposal to have a kind of a traffic light scheme. On the one hand, for example, the binding affinity is important, which is measured. Solubility is measured. Also the lipophilicity of the compounds, and there will be a number of predicted parameters like the molecular weight, polar surface area, rotatable bonds, and the fraction of sp3 hybridized carbon atoms, and the novelty, which will be measured in a certain way of the compounds. Next slide, please. So benefits of CASH. Participating it should be transparent and unbiased as good as possible, and the opportunities to benefit from open forum for academic and commercial participants, and to validate and demonstrate their capabilities. And for the community, of course, on the one hand, we'll make all the data publicly available for biologically interesting targets, and on the other hand, of course, hope to accelerate drug discovery by efficiently developing new computational methods. The next slide. So this is the homepage that Mathieu set up, cash-challenge.org. And next slide. And this is the paper that we recently published. Now, 1 week ago, we had also a publication. The detailed white paper was published in Nature Chemical Reviews. And I'd like to especially thank Christina, Al, and Mathieu. Without them, this would have not been possible. And of course, all the other authors here on the list who contributed substantially to the whole project. Next slide. Yeah, so short conclusions. So CASH is a public benchmarking project to compare and improve computational small molecule hit finding approaches with cycles of prediction, compound synthesis, and experimental testing. And I think that really the compounds will be synthesized and tested is a new aspect of CASH. And of course, we hope that it will enable the hit and identification method development for computational chemistry. So with that, I would like to give over to Mathieu.
Thanks, Alex, for passing the torch. I would like to start first quickly summarizing a pilot project that we ran at the SGC in Toronto over the past 18 months, where we partner with 2 biotechs that use AI for hit discovery. And we asked them to select up to 200 compounds for predefined WDR containing proteins, and we tested experimentally the compounds. So we decided to focus on WDR proteins. One of the reasons was that these are totally absent from training sets. There are very few known inhibitor ligands for this protein, and so we thought it was an interesting case study. Long story short, hits were confirmed for 4 out of 7 targets, and the 2 companies were Atomwise in San Francisco and Cyclica in Toronto. So they used their machine learning to screen against the crystal structure of the protein, millions of compounds, come up with 100 to 200 that were tested experimentally at the SGC in the Masoud Vedadi's group in Toronto. So here's the result for Cyclica. They identified hit for 1 out of 3 targets, and here is the hit. It's not as potent as one would have hoped, but this is a first in class for its target. It was very much reproducible in Alice Lee's hands in Masoud's group. And Sarah Kimani in Levon's group was able to crystallize the compound in complex with DCAF1. And then Julie Owen at Cyclica selected an additional 20 compounds to significantly improve the potency by SPR. Here are the results from Atomwise, identified, we believe, 3 hits for 3 out of 6 targets. Again, the hits are not very potent, but these are all first in class for their targets, and for a target class that is completely absent from training sets. At this point, we hope to see in the future SAR and crystal structure to further build confidence in these hits. So the next step we thought would be to open our experimental platform to all computational chemists and AI experts around the world, and this is CASH, which Alex just presented. And so here a quick summary on the first CASH challenge, which is focused on LRRK2. LRRK2 is the most commonly mutated gene in familial Parkinson's disease. There is a kinase domain in yellow here and the WDR domain in blue. And a number of pharma companies have developed ligands for the kinase domain that are in preclinical or early stage clinical trial. But I don't have time to enter into these details, but you can go back to it once the video is on YouTube. But about 16 or 18 months ago, this paper in Nature suggested that targeting the WDR domain may be actually an interesting alternative for this protein. So we are inviting people to focus to find hits for this WDR domain. So we received 35 applications. The call for application is closed now. We received 35 applications from a little bit all over, including top players in the field, top universities, 25 from academia, 7 biotech, 1 big pharma. And I can't get into the details of the methods that were provided. This will be released with the data at the end, but I can still give you a flavor. As you can imagine, many participants had at least 1 step of docking, high throughput, ultra-high throughput, consensus docking in their methodology. Many also had machine learning, deep learning docking, generative model, neural network scoring. There were some true and trusted refinement methodologies, steps, like molecular mechanics for energy perturbation. Also, many proposed to dock fragments at one step, or pharmacophore approaches, mapping water molecules using AlphaFold or Rosetta Fold to generate conformational ensembles. And a couple participants also said they would ask medicinal chemists to have a look at their selection. So very quickly, so for instance, one participant said, I'm going to have just 1 single step ultra high throughput docking, and the result will be what it is. And these 2 participants here said, I just have 1 step deep learning docking. This participant, well, I'll start with high throughput docking and I'll refine with deep learning docking. Well, these participants, I'll screen with deep learning docking and then I'll refine with high throughput docking. And so there were many such methods with 2 steps, 3 steps, 4 steps, 5 steps, all the way to 7 steps. Well, this participant here says, I'll dock fragments to generate interaction hotspots in my binding pockets, and then I'll use these hotspots to identify in the PDB similar pockets with a similar type of hotspots bound to compounds, and then the interaction between these compounds found in the PDB will be used to generate a pharmacophore hypothesis. I'll do a pharmacophore search. The output of the pharmacophore search will be used as a training set for deep learning generative model, and then the results will be refined with high-throughput docking. Well, whether this is genius or madness, I don't know. I guess we'll know in 20 months from now when the experimental data is in. But at any rate, I think it's going to be really interesting or fascinating to see how this type of very convoluted and sophisticated methods compare with more traditional approaches that we see currently in papers. A teaser, the next challenge will be focused on the helicase from SARS-CoV-2, more precisely the RNA binding site of this helicase, and there are a crystal structure in the PDB with fragments occupying these sites. The website here, if you want to know more, cache is a component of Target 2035, where International Federation of Biomedical Scientists from the public and private sectors are pursuing this mission to develop chemical tools of probes for all human druggable protein by year 2035. And this Federation of Scientists will not be able to achieve this goal in time with today's technology. So the first step is to identify technology to be more efficient about hit identification and hit optimization. And this is where we believe cache could be contributing. And finally, I'd like to acknowledge the hard work of my colleagues at SGC Toronto and partners at Cyclica and Atomwise who work on the WDR pilot study that was funded by Genome Canada. I'm underlining here people who did the actual work at the bench or at the computers. And these three companies here contributed actively in setting the framework for cache. And I'm going to stop here and thank you.
Thanks very much, Mathieu. So, I mean, we're a little over time, so we won't have any questions. But what I would like to say is, first of all, thanks to the speakers for giving us their time today and the benefit of their experience. And if any, and I'd like to also encourage people to communicate that this next round of cache challenge on this binding site in this helicase, which is the most conserved binding site in all of coronaviruses, it would be very interesting to get more participants. If you guys have let folks know that there's many ways to support the cache challenge, and contact Mathieu or Alex or Christina to let them know how you think you might be able to help, because I do think that this concerted effort of the community is the fastest way to get this kind of work going. And I would like to emphasize that the SGC will be committing to do the experiments, and the deal is the chemistry goes in the public domain for the world to benefit. And so I think that that's going to be a tremendous contribution to science and to drug discovery. And so with that,
and the generic description of the methods go in the public domain as well, right?
Absolutely, absolutely. So we really hope that it hits, you know, it's out of the park in two. The methods get better and the chemistry gets donated to the public good. And we'll do our best to choose cool protein targets that make it exciting for everybody to participate. And with that, I'll thank everyone for attending. Thank you for folks online. This thing has been recorded, so you can pass it on to your friends, and please participate in the next challenge if you think you want to have some fun. Okay, thank you so much. See you, everybody.